Let me get started. Hi, everyone. Welcome to our Momentum Academy AP Bio Open House. And guys, my name is Neha Mathur, and I will be the instructor for this course. Let me go ahead and go over a couple of things about myself. Guys, I have more than, 20 years of teaching experience, and I have been teaching the field of science, biology, and chemistry during all that time. I started my career as a biochemist. I have a master's in biochemistry, and I worked as a biochemist at Pfizer for some time. And then I also have a second master's in education. After that, I've been teaching in US for in private schools and public schools for the duration of my time here. Currently, I'm teaching at St. John's. I am teaching their honors biology courses, chemistry, and then I'm also doing a course called SRD, which is called Scientific Research and Design, where students are learning how to design an experiment and then conduct them. So, those are some of the things that I'm doing. That's where my whole idea of teaching curriculum is in a way that is more accessible to students because everyone is a different learner. So that's a little bit about me. Guys, let's go over the idea of the AP exam. This is an AP Bio prep course, which means we will review the course in 12 classes. And guys, that also means that we will cover content, but more importantly, we will do practice questions so that you can have an idea on the type of questions that you can expect. Guys, the class will be Saturday, 12 to 2 p.m. Central Time, and it'll start February 1st to April 26th. No class on March 8th, which is our spring break. Okay, so let me talk about the class. Guys, we are expecting that you have taken a pre-AP course, an honors course as a freshman or sophomore, and now you are expecting to take the AP exam in May. The whole idea of the AP course is application, but guys, application comes with knowledge. So, of course, a lot of our time will be spent reviewing concepts and then going over what type of questions can you expect on that content. Please do expect weekly homework, which is good because it's a review. In 2 hours, I will cover a lot of material. So it's always good that you can go back and review it through a homework assignment to make sure that you have solidified your understanding. About 45 to 90 minutes, guys. Sometimes it might be more. When I do assigned the free response questions, I will also give you a time limit where I want you to know that these are timed responses. And so you need to also prep yourself so you know that you don't run over the time limit. Guys, the AP exam itself is 3 hours long and consists of two sections, which is the multiple choice and grid-in questions and the free response. So it's kind of like equally distributed. So it's absolutely important that you understand the multiple choice, but there's a very specific way in which the free response question, or what we call as the essay questions, are graded. Guys, score distribution for these sciences for the last few years have been right here. And guys, as you can see, AP biology is very challenging to get a 4 or 5 on the test. As you can see, like 17 and 23%. And if you compare it with others, you can see it's a challenging course. And multiple things which I would say are challenging about this. Of course, the content, the breadth and depth of the knowledge that you're expected to know is a lot. And not only that, but there are some nuanced, like little things that you should definitely know to do well on the exam. And guys, that's what I would try to go over. If you're doing something, then what is important from the AP biology perspective? And that's what we will go more into. And the other thing is, of course, the wordings of the questions are pretty elaborate, where they will give you tons of different words. And so one of the things that you have to learn is how to find what's important, right? Well, how to go through the whole thing and know, okay, this is more important. This is what they are doing. You will expect multiple graphs and charts, and so learning them, how to apply your knowledge to them is also absolutely important. And so, guys, all those things make it a pretty challenging course to get a good score on. Again, not impossible, but there is a specific way that you have to study to do well on this exam. All right. So, guys, again, like I said, class description, it is an introductory college level class. The main idea is that you are done with the content, learning has already been done, right? The lower levels is what you are expected to do. But in AP Bio, it'll be the upper level application, inquiry, synthesis, those words where we kind of talk about whether you have this thing is knowledge, how do you apply it to graphs, charts, some sort of narrative. That is where this course will be more relevant. So any topic that we do study has to be in a form of inquiry. That means labs. Labs are an absolute important component of the AP Bio curriculum. And the experiments are also, the questions are also based on those inquiry investigation. Okay, so guys, the AP Bio exam, like I said, the structure is multiple choice section, 1 hour, 30 minutes, 60 questions, 50% of your score. And this is kind of a breakdown of the type of question, concept based. These are simple. Do you know the concept? Can you apply it? These are very simplistic type of questions. Most of the students get them. Visual representation, graphs, charts, setup of an experiment. This is where you also have to learn the application, those little things. What is an experiment trying to tell you? And then how do you connect it to the question or visual representation? Questions and methods. Methodology is mainly, again, experiment based. Data and representing and describing data. Again, guys, here we talk about charts and graphs. Statistical test, that is absolutely important in the last few years in AP Bio. And then argumentation. That means what tells you that this is the right choice. Again, that's the synthesis portion. So as you can see, the concept explanation isn't just one very, very small part of the whole curriculum. It's mainly understanding data, analyzing it, arguments for and against it, and then answering questions. And that is the multiple choice section. Once we come to the free response section, which is again 1 hour, 30 minutes, 50% of your total score, guys, this is where you need a lot of practice. And it depends from school to school. Some schools are really good, but it takes a certain way of understanding. AP Bio, they will give you a free response and they will highlight some words. And so understanding what does it mean? Justify, explain, define. Each of these words mean they're expecting something from you. Right? So the free response question consists of 2 long form free response. And guys, this is where most of the data analysis comes from. We also expect that this is where majority of your 1 hour, 30 minutes will be spent. And it's given like a expectation about 15 to 20 minutes on these 2 questions only. They also carry the most point values, so it makes sense that you spend time on them. And then after that, you have 4 short response questions, which require a paragraph length. And again, the point values go down. And so that's where you have these words like describe, explain. How is description different from an explanation? Predicting, justifying, representing. This is where you have to think what is required by you. And guys, every time we have a class, we will take time to go over these questions. We'll start from simple ones to what the most recent ones are and what are they expecting you to do that. right? So that's where we are going to talk about. AP exam date is May 5th, 2025. And so that's where we are trying to go, that we can finish in April. So that'll give you a long time to prep on your own. Okay, let's go on to the topics part, guys. Topics are, they build up. So we start with unit 1, which is chemistry of life, which is, we are biology, but the base of biology is chemistry. So it's beneficial if you have a basis of chemistry, if you've already done chemistry. And so you start with the idea of atoms and elements and compounds, and we kind of build up on that. But more importantly, we go over some very important molecules which are required for life, which is one of them is water. So this comes under the realm of inorganic chemistry. And you go over how does the structure of water creates its properties. Because guys, your properties of water. Water is absolutely essential, but what makes it so unique? There are so many compounds. Why water? So we'll go over how is it created, what does it do, its structure, and then how does it create these properties. So that's the concept part. But then yours is application. You will be given different scenarios and you have to see how what you learned can you apply to the scenario. Then we go to macromolecules, which is organic chemistry, carbon-based compounds. Two important things. Inorganic is water, second is carbon, right? Majority of the living things are made of these two things. In macromolecules, we'll talk about carbohydrates, lipids, and this is also, I feel, one of the challenging questions or units because of the depth of information and application that you are required. So now that once we have done about discuss chemistry, we go to the actual biology. And guys, that's where we go into cells. So see how we are building up on our information. And in cells, we talk about structure and function. So a lot of these cellular structure function you have done in a previous pre-AP course. So I'll be honest, you don't go much in depth in class. The expectation is you should know it or review it on your own. But now comes, so you know this, but now is the application, the why. Why does a structure look a certain way? One very important theme in AP Bio is structure related to function. Every time you study a concept, you need to study the why. Why does a molecule look a certain way? Right? So we go into cellular components, yes, which means what are the things inside the cells, the nucleus, the cell membrane, the other organelles, but why are they looking a certain way? Right? After billions of years of evolution, there has to be a reason why something looks a certain way. And so, guys, that's where the analysis portion comes in. So we talk about cell membrane, its structure, function. So as you can see, it's always structure, function. How is the structure connected to the function, right? And it builds up the structure. You need to know what is it made up of, and then why is it made, and how does it affect the environment. And then we also talk about, once we know the structure, how does the cell use it in its environment, right? Interaction with the environment, regulatory mechanisms like osmosis and selective permeability. So then we talk about cell transport. Things go in and out of a cell. A cell needs to survive. How does that happen? And how does the structure help the cell in its functionality? Right? Cellular compartmentalization is mainly referring to cell structures and how they are relevant. Guys, once we have talked about this idea, we go into cellular energetics, which is cell energy. Living things require energy to survive. And so this is where we talk about enzymes. Enzymes are absolutely essential for cells. Again, as you can see, structure, function. Again, enzyme is something, helps in the cell. Why and how does it do it? And then go to energy, two processes. We go over photosynthesis and cellular respiration. Now we know enough chemistry to go actually in depth into the steps of these two things. This is another unit where students struggle because of the depth of knowledge and the amount of information that you have to sustain, while making sure that you make those connections between the units that we were done already. Guys, after we talk about cellular energetics, we also talk about response to the environment. This is cell signaling. How does the cell communicate with other cells or along its environment? And that's where we go into cell communication and cell cycle. How does this communication work? And one of the ways by which cells communicate is cell division, when the cell has to divide. And so, why the cell has to divide? What are the different ways in which it can divide? And then, what will happen if these signaling things doesn't work correctly? Moving on, guys, from there, we talk about heredity, which is how are the cells passing on information, structure of DNA, RNA, genetics, inheritance, right? Mendelian, you guys studied about Mendel, but now we expect you to know more non-Mendelian, complex genetics, right? How does that work? And then apply it to your knowledge. And then finally, we come to gene expression. Guys, gene expression is so okay, we have genes, but how do the genes show themselves? What does that actually mean? And that's where we talk about very important thing called protein synthesis. How does the cell create these proteins, which you will see as a trait, right? And here we talk about genotype affects phenotype, mutation, genetic diversity. And then the last component of this unit is genetic engineering and biotechnology. So, guys, biotechnology is a very interesting unit, and AP Bio is adding information to it constantly because we are constantly finding new things. So, in genetic engineering, we learn how we manipulate genes, and then we use the techniques. And usually, you will do a lab in the school. By the way, every unit has labs with it because the lab unit is what you will learn the application of. Then, guys, our next unit is natural selection. Natural selection is evolution. So now that's where we are building up to. Evolution works on organisms, how it works, right? We talked about Darwin, which is probably you might have studied in the pre-AP course, but now we will go more higher. We will try to connect genetics to evolution. And that's where we talk about how species are changing over time, what are extinction, what are phylogenetic trees, which is kind of like a tree line of how different species have originated. And then origin of life on earth, like we studied all this thing, but how did life actually started? What were some experiments? And finally, guys, we go to this big picture of ecology. Now we have living things, they are all interacting with one another. How do they affect one another, right? So if you see, we are starting at the atom molecular level and we are moving up to have this huge environment and how it affects us, right? So as you can see, they are going from smallest to the most widest, but you have to keep track of how they are connected with one another and what will happen if one thing is not doing its job correctly, because it's like a pretty big pyramidal, it's a hierarchy that's going up. And what will happen if something doesn't do its job, while also making sure that you can apply all that information. So as you can see, guys, that's a pretty long order, right? It's a lot of information that you have to keep track of while also making sure that you understand the connections between them. Okay, so let's go to what we will be doing in the class. First of all, guys, we will use Zoom. It would be an online platform. You guys will get a Zoom ID that will help you join the class, which will be sent to you. And we will use Canvas as our platform for classwork, homework, and everything. So let's go over our platform. So this is what your platform will look like. Guys, once you'll go, you will have some setup and I'll show you some of the things, but Canvas is what we will be using to sharing class material. All the all the slides that I use, I will upload them ahead of time. Homework, worksheets, all of that will be available to you. Weekly homework, you will have a place that you can submit it. It needs to be submitted by the next class. But guys, this is an AP class, so what I expect from you is before every class, I'll give you some time. We will review. We have to because it builds up. But if when you were doing the homework and you had questions, I want you to clarify them before we move on, right? All the class material you will be available to you for at least 3 more months from the last class, which will be more than enough for you to take your AP exam and go on from there. Guys, one of the other important things that we do is that we will be recording our classes. So let's say, I'm starting this class in February 1st and you are taking the exam in May. Even though we reviewed, you forgot. No worries. Recordings will be available to you. You can go back, check the recordings. How did we do a certain type of question? What was the main thing that you need to know about a topic? That will all be available to you. Okay, so guys, from there, we will go into some other things. But guys, before we go, I wanted to show you a little bit about the Canvas setup. So guys, this is what the canvas will look like. Now, of course, what you will see will be much more different. You're seeing the teacher's view. So that's different. But guys, this is the most important thing, modules, right? And this is where your class will be set up. So these are some of the important reference material that you will be using. The objective courses, that means at each unit, AP Biology requires you to know something very specific about that objective. So we'll go, it's available to you if you want to go over that. This is the AP Biology equations and formula sheet PDF. Again, guys, the PDF it has all the formulas that you need to know. So we don't expect you to learn all of that. It will be given to you because every unit has some sort of math component in there. So the formulas will be available. So this is what it looks like. We will start with statistical analysis. That's usually our first unit. And so all the things that you need, the formula is there, but the application is what you have to learn. So everything that you need is right here given into this formula. But every formula is connected to a certain unit. Okay. All right. FRQ tips and terms. Again, we will discuss that in class. When you are taking FRQ, what are some of the things that you need to keep in mind, right? Because they have a very specific format of asking questions and a specific way that they want you to answer that, right? So we'll go over those, like what does that mean? So for example, if they say compare, that's easy. We all know compare means examining two or more objects and seeing what are the similarities. Contrast is differences. Identify, just name. Identify. Explain. Now, this is where they want you to be a little bit more descriptive, right? Give some reasons. And whenever they use the word explain, there might be like 2 points associated with it because first you have to say what the term is and then explain or describe what it means, right? Describe, give a picture or a count of the words, right? Describe 2 or 3 of those. Discuss different points of view. Define, justify, analyze, apply. So guys, they are giving you some very, very specific words. Evaluate, judge, examine. You have a data, something else is given, evaluate the conclusion. Is that works or not, right? Write in a paragraph form. never bulletin points. You can, guys, it's not like you can't use it, you can, but just bulletin points themselves are not good enough. You have to have some sort of descriptor to go with that, right? So, again, don't worry about perfect grammar. This is not AP line. But of course, vocabulary words, you need to make sure that you're writing them correctly, right? So, those are just some things that we will be talking about when we talk about these things on the AP Bio exam. Graphs are huge, like for every FRQ, at least the first two FRQs, you will definitely get graphs. So that's a big thing. You would be expected to read a graph and analyze it, and you would be expected to make a graph, both of them. So we will go over if you have a given template and you have a data point or a data table, how do you decide which graph are you going to use? That's huge in there. And once you use a graph, then you have to analyze it and then answer questions on it, right? So that's where graph choices, and these are the different graph choices that you have. You would be expected to know what's a histogram, what are box plots, dot plots, bar graph, scatter plots, line graph, pie chart, stack bar. All these are different forms of visuals that you can see. And you also have to know why you are using a certain, what is the advantage, what does the data look like, so you can use a certain type of graph that goes with it. All right, guys. So those were just some things. Then we come to every week, guys. Every week when I'll open it, you will see the objective. What are we doing in week one? We are going to discuss scientific method and statistics, which is huge for everything that you are going to do. You will have slideshows, so you can take a look at the slideshow. All these slideshows will be available to you. So don't worry about, oh, I don't have that. But everything that I do will be provided to you. And these slideshows are pretty easy. You can go over them. Practice, guys, that's where we will be spending a lot of time. Yes, I'll do content, but then most of the time that I want to spend is on practicing, right? Because content you should have covered in school. Also, I'm there more for clarification or going over, okay, this is important. This is what you need to know. But then, guys, stats practice is where we are going to go over. So we have tons of practice sheets. Guys, I'll be honest, we don't have enough time for me to cover content and practice. So I'll do a couple of practice questions with you just so that you can get an idea how to do the question. But these practice sheets are pretty long. So the expectation is that you will finish it on your own. And then the next time when we meet, you can go ahead and ask me questions. Now, how do you know whether you did it right or not? Guys, I will also always attach a key to it. That key will not be open in class, but I will publish all of them at the end of the class. Again, the expectation is that you do these questions on your own, check your work, and if anything is missing or you didn't get it right or you still have questions, then you ask me about them in the next time. We'll also do FRQs, like I said, really, really important. So, guys, once we do an FRQ, I'll also put the key. Guys, what does key mean? So, we use a rubric to grade these FRQs, right? Which means your AP reader is looking for certain catchwords, right? And that's what we are going to go over. So, we'll do at least one question in class. And the questions usually has multiple points, right? Let me show you an FRQ question. It's taking a little bit to open the documents. But guys, each FRQ question, and you might have done this in class, is usually a big blurb, some sort of visual representation that goes with it, and then tons of sub questions. So we'll do a couple of questions in class, but then again, the rest I expect you to finish on your own. I will upload the rubric. I do want you to take a look at it and see what you wrote and what would be a perfect answer. And was there any differences, right? And why you missed those differences? Was it because you didn't know the content, or you didn't understand that's what the question was asking? Like, we need to analyze, identify what's missing, and then we can go from there, right? Again, guys, sorry, it's taking a lot of time, so I'm going to move on. But that's how every week would be. 2 hours, guys, that's what we are going to do. And then you will have some quiz. Again, it's a homework grade. I'm not here to kind of tell you, oh, you didn't do this. I'm not here to grade you, but I'm here to identify what could you have done better to improve, right? So some more practice. And then, of course, all the recordings will also be connected over here. Same thing, guys, week 2, same format. You know what we are doing. Slideshows, homeworks, 3, 4, and every week you would see the same kind of format. Okay, guys, before I move on, any questions? And again, guys, while I'm talking and something pops in your mind, please put it in the chat or use the, raise your hand emoji, anything, and I would be more than happy to answer questions. So, guys, the last thing I wanted to do was let's take a look at some sample questions and see what would you do to answer them, right? So I wanted to do some multiple choice questions and then an FRQ, right? Just so we have an idea. And again, guys, don't worry about, oh, I don't know the content. That's okay. I just wanted to give you an idea on what to expect and how to go about doing it, right? Remember, 2 parts, multiple choice and FRQ. So let's take a look at this question. I also wanted to show you what do you expect when you are in class. So guys, I write a lot because there's a lot of content that we have to go over and I have to explain a lot of things. So I like writing. And this is what I do. This handy dandy little whiteboard that I like to write on. Guys, when you are taking this class, what I would like to do is pin my window. I hope you guys know how to pin. There are these little 3 dots. Click on them and then you pin. And when you pin that part, then that becomes the bigger picture rather than you seeing that in a small, small window. It becomes the biggest window for you, right? So pin my window so you can see it better because I write a lot. Again, I go a little fast, but if you have questions, just stop me. I'll be more than happy to go back and explain. Okay, so guys, let's take a look at a simple multiple choice section question. So the question says, a common laboratory investigation involves putting a solution of starch and glucose into a bag and suspending the bag in a beaker as shown. So one thing, guys, that I always say is there are words, at least in multiple choice section, you won't have that many words, but the visuals are important, right? So let's take a look. Let's not even think about the question or the options. Let's just take a look at the visuals, right? So looking at the visual, I would even say, let's kind of make like a rough draft of what you are seeing and what is the picture actually showing you. So I have something like a bag. Again, guys, don't care what dialysis tubing is. If you know, great, because it's actually an experiment that you do in class, which includes dialysis tubing. But if you don't know, it's okay. But there is a bag. And inside the bag, you have starch and glucose, right? And then outside, I have tons of water. So what it's showing me is that I've suspended a bag filled with starch and glucose, and I've suspended it in water. Okay, that's my starting. Let's take a look at what happens over time. So first thing, I see that the bag is a little inflated, right? Which tells me something definitely should have gone in, right? These are the visual clues that you're supposed to see. Okay, let's take a look. What is my labeling showing me? So now my labeling is showing me that there is starch and glucose inside, which makes sense because it was before also. But now I see that there is glucose and water inside. So, guys, now I'm going to pause because something happened from my initial setup to later, right? And I'm not even looking at the questions right now. I'm going to make kind of like a mental picture of what has happened. And I already know that the water is a little inflated. That means something moved in, and what was outside is water. So, one thing is maybe the water moved in the bag. Makes sense because the bag seems a little inflated. Second, there was starch and glucose in, but then I saw glucose outside. So, hey, did the glucose move outside? Seems so. And my third component was starch. And guys, starch seemed to stay in the bag, right? Okay, this is the lab setup. Now, I also want you to think about how is this connected to content? Because, guys, that could be one of the questions. And so, this is something that we talk about when we do cell transport. When a cell moves things in and out of a cell, and guys, there are some very important criteria that controls what can move in and out of the cell. So we will do the content, and this is where this is connected. The only difference, and this is important, this is not a cell, it's a dialysis bag. So it's a simulated cell. So it might not be exactly like that, but it would be somewhat like that. All right, so now I have made some idea. Let's look at the option. The investigation is aimed at understanding how molecular size, now that's important, guys. They are talking about the size of the molecule. That means that is what's affecting this movement. So now, before I even go forward, which molecules? Water, glucose, starch. How the size of the molecule affects movement through a membrane. Okay, I already made some conclusions. Water moved. If it moved, that means it's small enough to move because things small can pass through. Glucose also moved. That means that would also be smaller. Starch, I know, did not move. That means size wise, it could be pretty big. And guys, this is where your previous unit comes in. Before we do cellular transport, we have already done biochemistry, where you know water is a very small molecule, inorganic H2O, right? That's where size kicks in. Glucose is a monomer. It's a bigger molecule. In fact, the formula is C6H12O6, so definitely bigger than water. But guys, starch is a polymer. This is when there are multiple glucose attached. It's a long thousands of chains of glucose. And so, yes, it does make sense. Size wise, water is the smallest, glucose becomes a little bigger, and starch is the biggest. And so, it kind of definitely correlates to what I know, to what I see in the lab. Okay. Which of the following best represents the amount of starch, water, and glucose in the dialysis bag? Guys, this is important. They are talking about what's happening in the bag over the course of the investigation. Okay, so now I have a pretty good idea. I know that in the dialysis, my water moved in, right? So it went up. Starch did not move, so it should have been consistent. And glucose, it moved out, right? I already know all of this before I see the options. Okay, guys, now in your chat, tell me what should be the correct option between A, B, C, and D. Go ahead, look at the graphs. And put in the chat, what do you think would be the right answer? Guys, the reason why I'm doing it because this is exactly how it would be in the classroom. right? That's how I can make sure that we are working together. So in the chat, A, B, C, D, what would be the correct answer? Okay, so I see some options. So in the first one, I'm seeing water, which is this dotted line in the bag. It has gone up. The dotted line is starch, which is kind of staying the same. And the glucose, which is the solid line, is going down. Guys, this might be the right answer, right? But let's see why the others won't be correct. So in B, I'm seeing water, which is the dotted line going down. Guys, that's not what our conclusion was because the bag inflated. The starch remained consistent. So this could also be true because starch did remain consistent and glucose went up. Guys, that's not true because I know glucose was outside, right? The other two are definitely not the option because it's showing you how the starch is changing, right? Now, all this, I took about 5 minutes to explain it. You have about 2 minutes to make all of this inference, right? That will give you enough time for you to read the question, make sense of it, answer, and move on within the time limit, right? But guys, that's what we got to do. We got to know the content and we got to know what is it that I'm looking for to answer these questions. Okay, let's move on. Let's see FRQ question, right? So this is a simple, and I say simple because it's an older FRQ question. And guys, we'll start with something very simplistic, which is scientific method. So we all know scientific method, right? And in scientific method, we talk about experimental design. How does the variables affect? And guys, we talk about the independent variable, dependent variable, control, constant, right? All these things we talk about. And so that's where this is. So something like this would be your FRQ 1 and 2. That means the first question would be something like this. Oh, sorry, this is actually the third experiment, third question. But even more would be the first two. Again, let's take a look at how to go about doing an FRQ. Same thing. I'm going to read the blurb and keep writing important things, right? Underlining if you are doing it. Seagrasses are aquatic plants that reproduce sexually. Male seagrass flowers produce sticky pollen. So males produce pollen, right? It's sexual reproduction because, guys, sexual reproduction is one of the cell division meiosis that we talk about, and you do have gametes in there, so that makes sense. Males have sticky pollen that is carried by water to the female flowers, resulting in fertilization. So the female plants we know has eggs. That means fertilization happens. So it helps if you kind of like make like a flow chart so that you can have an idea for yourself. Because guys, this is actually a small kind of like a uh, body of the paragraph. They are usually much more intense. A researcher claims that mobile aquatic invertebrates can also transfer pollen. Now, here's the thing. I don't know what does that mean. Mobile, aquatic, invertebrates. So mobile, I know, something that can move. Aquatic, I know, lives in water. I don't know what invertebrate is. It does not matter. That does not matter. What, let's say I say A, something. So the researcher claims something else can also transfer pollen from male to female flowers. So the claim is that instead of the circulating water, something else can also transfer pollen in the absence of circulating water. To investigate this claim, the researcher sets up an aquarium to model the possible interaction between the invertebrates and seagrass. So now to test the hypothesis, and the hypothesis is that if there is no water, these invertebrates can also help in fertilization. That's the hypothesis. Use the symbol below and template aquariums to demonstrate the experimental design to claim that mobile aquatic invertebrates can pollinate seagrass and the absence of circulating water. Draw the appropriate symbols in the negative control aquarium. So guys, this is where your concepts come in. So when we talk about control, control is something that you use to compare. A negative control and a positive control are different, right? So this is where your concepts come in. A negative control, and there is something called as a positive control, which we also called as the experimental set. So they are saying that I'm giving you a template for a negative and experimental aquarium, and you have to use these symbols to show how they will set up, right? That's what they're asking. So guys, here you can only answer the question if first you know what is a negative and experimental thing, then only then you would know. But guys, a negative control is the control, it's a true control. It's the one that does not have the independent variable. And in our case, the independent variable were the invertebrates, because that's what the scientists want to find, right? So in my negative control, I will do male and female. But in my experimental, I will do all of them along with the invertebrate, right? They say do not use any symbol more than once in the same aquarium. That means don't make tons of circles or squares. Just leave it as is. So again, looks easy, but guys, you need to know content, right? So this is the foundation, but then now you're applying it. Okay, identify the dependent variable in the experiment, predict the experimental results. And so guys, that's how we do these kind of type of questions that we are going in, right? And then after every one, I will give the rubric to you. So the rubric will look something like this, where once I've given you time, and guys, when we'll do these FRQ questions, I'll tell you, hey, let's simulate the same things as an AP exam. So for this question, I don't want you to use more than 5 minutes. You take 5 minutes, you do the best you can, and then we discuss. And once we discuss, I'll open up the rubric and we'll be like, okay, give yourself these points. Did you get all the points? And if you didn't, what were you that you were missing? Right? So guys, and again, I just wanted to show you because I want to see how the whole setup of the class will be like. It'll be a very interactive class. I ask a lot of questions. So I do expect you to have your videos on. You can either unmute yourself to answer the question or put it in the chat, but it has to be pretty interactive for me to see whether everyone is understanding or not. Otherwise, guys, it's just like a lecture. It gets too boring. So it has to be like a given forth to make it interesting. So, guys, that was something that I wanted to talk to you about. Any questions? All right. So, guys, it was really good talking to you all. Please know this is through Momentum Learning. And guys, here are their information that you can get to. And I hope that you will take the class. And I hope I'll see some of you. If you have any questions, you can stay back. But guys, that's all from me. And I will see you all 1st February. Bye, everyone.